So to get started, the origins of the U.S. vaccine movement, it was a collaboration between an evangelical minister and an African slave. What we're going to cover today is going to involve the vaccine strategy in the United States and other developed nations, starting with the battle against smallpox. We're also going to touch on the scourge of smallpox and the breakthrough collaboration between Onesimus and Cotton Mather. Does anybody know who Onesimus is? Okay, you're going to learn some great stuff today. We're also going to talk about some vaccine strategies used today, covering all the different types of vaccines. We're going to cover the vaccine studies that are going on right now in our community here in Northeast Florida and South Georgia. And then we'll have some time for questions at the end. And with that, I'm going to turn over the clicker to Dr. Helow.
Thank you. What does that mean? It goes forward?
You have the power.
All right. Here we go. So smallpox. She already told you that we were going to talk about smallpox and why was it such a big deal? Well, first of all, it was something that's been around since the Egyptians. They found it in mummies, and it's a horrible disease. They not only found it in the Egyptian mummies, but one of the big issues was that it was able to get around once we began traveling around from Japan to China, China to Japan. You can see the different centuries that those different things happened, including the slave ships that came to the New World in the 16th century. You look like you're going to say something. No.
I'm all ears.
And then in those early days, 3 out of every 10 people who got smallpox died. And in the 1900s alone, there were 300 million people who died from smallpox.
Yeah, I think the only thing worth noting is that when we talk about pandemics, the mortality rate of smallpox was so much more than the mortality rate of COVID-19.
Yes.
So COVID-19, you're looking at a mortality rate that's probably 1 or 2% in the U.S. compared to 33% for smallpox back then.
And it is a virus, which a lot of these things are, including COVID. It's a brick-shaped DNA virus, which what that means is, you saw it on the last picture. It's just kind of interesting. Not a lot of viruses are shaped like a brick. Normally you see them round, as you see in the media. And it causes this horrifying disease, as you can see, all the pox all over this individual. And these pox not only occur on the skin, but these pox swellings can go into any orifice, including the mouth and the nose and the ears and more importantly, down into the lungs. And that was generally the cause of death. People would get so much inflammation in their lungs, they would literally suffocate to death because of these pox being in the lungs. So pretty horrifying, as we know, even with the COVID coronavirus, we're having that same shortness of breath feeling. And so fortunately, there have not been any cases since 1978. It's been officially eradicated due, of course, to what we're talking about, vaccines. It was the World Health Organization declared it eradicated from the earth as of 1980. There are currently 2 labs that do still have it to be able to study it. And other than that, it's not found anywhere in the world. And so he's going to talk a little bit about the outbreaks and the history in North America of smallpox, and this is quite fascinating. Hold on.
Yeah, thank you, Victoria. Always a pleasure to work with you. Thank you for coming, everybody. And yes, I am the local historian, so I actually love to lecture about history. So this is my opportunity to do that. I like talking about medicine, but history is a lot of fun. So here's my opportunity to share with you some neat things. Before I get to that, I'm just kind of curious, by a show of hands, who has heard about this concept of Onesimus and Cotton Mather working together, an evangelical minister and an African slave? Has anybody heard of that? Who's not heard of it before? Okay, and who's skeptical that it ever even happened? A couple. Okay, so we'll address all of those things. So first of all, as Vicki mentioned, smallpox was just an absolutely devastating illness. And it came in cycles. There would be years where there was a pandemic year that literally 1/3 of people in a community were wiped out by smallpox. So you can imagine how frightening it must have been for people back in the day. And that's when they really didn't understand what we understand about viruses and the way infectious disease works. So it was incredibly intimidating idea. And if they heard of the fact that there was an outbreak coming, they were really, really frightened. And this little advertisement shows you how important that was in the mindset of people back in that day. So when slave ships came, people had the understanding that this disease was transmitted by people or something on the slave ship. They weren't even sure if it was people or rats or whatever it was on the slave ship. But they knew that that's where it came to a new community. And as Vicki mentioned, that was not new. That was not something related to the Americas. That probably happened back in ancient Egyptian days. We know that the Chinese brought it to the Japanese back in the 6th century. We knew that Arab traders that roamed around the world back in in the 11th, 12th, and 13th century were spreading smallpox, and the Portuguese brought it to Africa, and then ultimately it went from Africa to the Caribbean and to the New World. So this was well known at that time. It was so important to people that they actually had advertisements that if you were going to purchase slaves from a slave ship, that you wanted to know that the people on the slave ship either had smallpox before, because there was this understanding that once you had smallpox, chances are you wouldn't get it again, or they had been quarantined. So this was a huge part of the culture and the commerce of moving people from one place to another back in the 14 and the 1500s. The pandemics that came would typically start, again, with a ship. And it turned out that this was important in the way wars were fought and also what happened to the Native American population. So there's a lot of discussion about what happened to Native Americans who, by the way, they came from elsewhere. Native Americans migrated from Asia across land bridges that were frozen between Russia and Alaska back 10,000, 12,000 years ago. They populated the Americas, but when the Europeans came, a lot of them got sick from the diseases that the Europeans brought. And for that reason, the Native Americans became depopulated. And certainly the communities that were in the United States of all different races were prone to these infectious disease outbreaks. And it turned out that in 1721, it became known in the colonies in North America that it was a pandemic year, that there was an outbreak in London, and that outbreak was something that could eventually get into the colonies through ships that were constantly going between the different cities on the eastern seaboard of North America and the UK. So one of the things that we like to train our physicians about is what a clinical trial is. And the first clinical trial was actually conducted in Great Britain back in 1721. And what happened at that time is that people, the intelligentsia of Great Britain, the people who were a part of the royalty class, knew about how horrible smallpox was, but they also knew about the fact that people in the Middle East and Africa had come up with this idea that if you take the pus of somebody that has smallpox and you put that pus in an incision, that seemed to protect people from getting severe cases of smallpox. So just think about that. Just imagine that for a second. You're going to take the pus of somebody who's sick, who has a 33% chance of dying, and put it into a cut that's made by a surgeon with very crude instruments. And so the fact that
antibiotics.
Right. No. So the fact that people were even willing to do that is a testimony to how afraid they were about smallpox, but also the fact that they were desperate for any solution. And this was called variolation. And that was the technique of giving the infectious disease through a different route than you would get it ordinarily. So ordinarily you would get smallpox in through the respiratory route, but if you gave it through the skin route, your immune system would kick in before you died of the disease. And you'd have a mild case of disease, and you would be protected from subsequent outbreaks. So this was first just being discussed amongst people that were in the educated classes in Great Britain and in the United States. Before it was the United States, it was still the colonies then, just to remind everybody. And a small clinical trial was done in the Newgate prison in London, which was the horrible prison. It was a place where the rats didn't even want to go because it was so dirty, just to give you an idea how horrible it was. And there were 6 people that volunteered for a clinical trial with the promise from the King of England that if they participated in the clinical trial, they would be freed of their sentences, which were often death sentences. So
Death sentences for silly things like stealing $10 worth of something. And it was the 6 inmates were very low-level criminals, but they were sentenced to death. So that was very interesting when you read the biographical information on them.
So anyway, this was a structured clinical trial. And we call it the first clinical trial because there was a plan, there was a protocol that was put forth, and these 6 people were chosen. The king signed the consent for them. Obviously nowadays we go through this long consent process, but back in those days, the king was the one that gave consent. which, by the way, is still the case for the military. Did you know that the president can give consent for a soldier to participate in a clinical trial? So that may have changed a little bit less than you think, but in all the clinical trials that we run, every patient goes through a very long and thorough consent process.
Well, but the military person consented to give that president the consent.
Okay, well, that's a technicality. We'll go with that for now. But the point being is back in those days, it was the king that gave the consent. There were 6 people that participated in the study. And interestingly is that all 6 of the inoculated subjects remained free of smallpox during this horrible pandemic that occurred in 1721, including a couple of people that were exposed to patients with smallpox. In fact, one of the women that participated was paid to sleep in the same bed as a child with smallpox, and she never got sick because she was inoculated. The other thing that was interesting is that one of the volunteers did not disclose the fact that he had been infected with smallpox previously. So it was known that he was protected based on the fact that he survived it already, but he did not disclose that so that he can get off his sentence, so he can have his sentence commuted and get out of prison. So
Yeah,
that was pretty clever on his part, but we would call that a protocol violation in the research world.
Not telling the truth.
That's right. So obviously it's important that when people participate in clinical trials, we know really what happened in your past medical history and understand these things. And if it doesn't happen, then we have an issue. We'll get to questions in a second, but go ahead. Well, he probably eventually admitted to it. Yeah, it was, obviously things had a way of coming out.
Yeah, when I read the details about it, he did admit to it because actually they tried to give him a second dose, and they ran out of it to give all of these 6 prisoners some dose, and he stood up as, well, I already had the disease, so I don't need the second round.
Yeah, so that's actually, it's actually very interesting, is because he had already survived smallpox, they couldn't even get a good infection in his skin because his immune system was already protecting him. So he was different than all the other subjects. They put the pus in, and they had this big inflammatory process. But him, it didn't happen because his immune system was already protecting him. So he fessed up because of that.
Right.
So you're right. We didn't have a biological test to show that, but we actually had some clinical evidence to show it. So back then, they had ways of figuring things out also. So the other thing that's interesting is that 3 of the 6 prisoners who were involved in these clinical trials, who were released from prison, ended up back in jail for other crimes. So it shows that the scientific world may be better than the social justice world.
Right. And of interest is these prisoners were 19 to about 23 years of age. And 3 of them end up back in prison, and again, for low-level crimes, but they were punishable by death crimes.
So on the heels of that, there was this experiment that occurred in the Americas. And this experiment was a unique collaboration, as we mentioned, between an African slave named Onesimus and a Puritan leader named Cotton Mather. And this was triggered by the fact that a ship came. It was a British ship called the HMS Seahorse, and it arrived in April of 1721. And then during the trip, one of the sailors began to exhibit symptoms of smallpox. So when the ship took off, they didn't know anybody had smallpox, but by the time it got to the New World, the disease had spread in the ship and then ultimately in Boston, despite the fact that the sailor was isolated. So people were freaking out, you can imagine. So at that time, there was a Puritan leader named Cotton Mather, who was the son of another very well-known Puritan leader named Increase Mather. And Cotton Mather was a very intellectually curious person, and they were talking about the fact that they had this horrible disease that they had to deal with and what strategies. And Onesimus, which was a slave that he had, was actually given to him by his congregation some years before, who he actually enjoyed talking with. Cotton Mather noted that Onesimus was a very, very intelligent guy and somebody that he could have lively conversations with. And Onesimus mentioned to Cotton Mather that he had been inoculated against smallpox as a child and showed him the scar from the inoculation, and that got Cotton Mather thinking. And it turned into this crazy collaboration. So, but before I get to that, I just want to give you some sense for how effective this concept was. So if you look at this graph, and I'll walk you through it, these are the years of the different smallpox pandemics in the New World. And this is data from Boston. So the one that I'm going to talk about in detail is from 1721. So if you look at the 1702 and the 1721 pandemics, you see that there was between a 5 and 8% mortality rate in Boston. So again, this is over the course of a few months, but during those few months, between 5 and 8% of the people in Boston died of smallpox. So just let that sink in for a second. And then they started this practice of inoculation, and then it started working, and during subsequent outbreaks, and this is well before you had a formal vaccine, but during subsequent outbreaks, this, quote, folk medicine remedy of inoculating people reduced the mortality rate down to 1%. So this was a really, really effective way of changing outcomes related to smallpox through this collaboration. So let's learn a little bit more about Onesimus and Cotton Mather. So Cotton Mather, he was a Puritan minister and a very prolific author, and he wrote something called Magnalia Christi Americana, which translates into something like the great deeds of Christ in America. Mm-hmm. So he was a minister. These were things that came out of religious fervor. He believed that, like a lot of educated people at that time, particularly educated ministers, is that studying science was trying to understand how God works. And so he was really focused on doing botany experiments and doing other things. So when he learned about this inoculation concept, it fit right into his way of thinking. Onesimus was somebody of unknown birth name, and he is now somebody that we all owe a debt of gratitude for. Now, Cotton Mather got united with Onesimus back in 1706, which was about 15 years before they actually did this project together. And Cotton Mather actually enjoyed his interactions with Onesimus and commented on multiple occasions that he was a very intelligent person. Onesimus was also frustrating to Cotton Mather, as an aside, because Onesimus would never convert to Christianity.
Right.
And so throughout their interaction, Cotton Mather was trying to get him to convert, and he never did. And eventually they broke up their, quote, partnership, and Onesimus went off to somebody else after maybe 20 years because Cotton Mather was unsuccessful at converting him.
And I'm not converting you.
Well, that's another story. Any event, Cotton Mather promoted the new Newtonian science in America and sent many scientific reports to the Royal Society of London, and they formally elected him as a fellow in 1723, in large part because of this study on inoculation. And I'll show you some of the details of that. But what's interesting about Cotton Mather is that he really wanted the approval of the intellectual community. And so he was really, really excited about the fact that the Newtonian Society in the UK elected him as a fellow.
Talk about his wig.
We'll get to that in a second.
Okay.
So in any event, as I mentioned, Onesimus introduced Mather to the principle and procedures of inoculation to prevent disease. And again, this is the foundation of vaccines. What is a vaccine? Well, basically a vaccine is introducing an antigen, the bad protein, to your immune system in a dose that your immune system can handle so that you're protected against the disease in the future. That's what vaccines do. And so this was the first major experiment that looked at this concept and showed that it actually worked. And just a couple of things that I think are pretty interesting is that Cotton Mather was actually very controversial in his day, in part because he was involved in the Salem witch trials. And at the time of the Salem witch trials, he was an attorney that was actually arguing that the women who were having dreams, bad dreams, were actually witches. So it was interesting. He had a mixed track record. He was probably partially responsible for a lot of these young women being executed as witches. But at the same time, he said that if you use dreams as evidence in court, that's probably not valid. So he was considered a little bit out there. Really what he wanted to do was to become the president of Harvard College, but he never got there. His dad was, but they felt he was a little bit too controversial to become president of Harvard College. So another little interesting aside. Onesimus is, although he came over to this country as a man without even knowing his birth name, he's now a very famous person, a very influential person, and was voted by Boston Magazine as one of the best Bostonians of all time. So really a testimony to his contribution to what we do now in terms of inoculations and vaccines. So these are the actual data from the study. So Cotton Mather, he wasn't even a physician. He was basically, they used to call them patricians, people that were just smart in different ways. And his primary role was an evangelical minister. And he recruited this guy named Dr. Boylston, And they inoculated 287 people in Boston in 1721, and they just followed their outcomes. So it was a much bigger study than the one that was done in the UK, and it had much more community outreach than the one in the UK. So instead of working with people in prisons, they actually worked with people in the community, which is a whole different way of looking at it, and it's the American tradition versus the European tradition. And so you can see here that when you look at the percentage of the population that got smallpox naturally acquired versus getting cases of inoculated smallpox, that back in 1721, there were very few people, only the Cotton Mather people and the Nesmus people that were getting inoculated. But during the course of subsequent outbreaks, a greater percentage of people got inoculations and fewer people got smallpox or natural infections. And look what happened with the percent of population that died. This is actually the death rate. And as I mentioned, in the 1721 pandemic, 8% of people in Boston died of smallpox. But as people are getting inoculated, it went down and down and down until it was about 1%. And remind you that the formal vaccine that was developed by Jenner in the UK, which was actually cowpox, didn't come out until 1800. So just this natural remedy had reduced the mortality rate tremendously over the course of about 80 years. So really quite impressive data. And this is just a good picture of, this is an old Shutterstock picture of two boys, same age. One was inoculated and one was not. And see how they respond when they are confronted with the disease. These were both boys that were exposed to the disease, but one was protected and the other one was not, and you can see the difference. So I think it's a great example of what we just talked about in terms of what we call cross-cultural magic. And there's a lot of examples of that in science and in medicine, is that when people from different backgrounds and different perspectives get together, they come up with a solution that's better than any of them would have come up with individually. Mm-hmm. And I like to call this cross-cultural magic. And so another example of this is actually with the COVID-19 vaccine. It has a little bit of a personal connection for me because of this fellow named Dr. Drew Weissman. Has anybody heard of him before? Yeah, you probably wouldn't hear of him, but I happen to know him because I went to college with him. He was in my college class. And we actually were both chemistry majors, so I took a lot of classes with him. And he was a nice guy, kind of quiet guy. No one would necessarily thought he was going to be a superstar. But he will probably win the Nobel Prize in medicine for what he's done for the COVID-19 vaccine. So it's kind of interesting from my perspective to have associated with him during my very early years. So just a little bit about his background is that he went to Boston University Medical School, and then he became very interested in working on solutions for HIV. And he wondered if messenger RNA could be a solution for HIV. So he did work in Dr. Fauci's lab back in the 1990s, and he started to study the immune responses to different foreign proteins in an effort to try to create an HIV vaccine. And then he met the woman here you see here, Dr. Carrico, who was from Hungary. And she actually grew up during the communist days of Hungary behind the Iron Curtain, and she was somebody that grew up on a farm. It was a two-room home that she grew up in with very working class people or her parents. I think her dad was a butcher and her mom was a bookkeeper. And she studied in their little school there and eventually got invited to come to the United States to be a graduate student. She was a graduate student at Temple University and then ultimately took a job at the University of Pennsylvania in 1989 where she met Dr. Weissman. And one of the interesting parts of her story is that she was quite poor and she had to kind of get out of communist Hungary at that time, and they didn't allow anybody to bring currency outside of their of the country because there was such a flight of capital in those days. So in order for her to bring enough cash to survive the trip and to buy basic goods before her first paycheck, she actually smuggled money in a teddy bear when she came to the United States so that she can get money out of Hungary for her basic needs. So it's kind of a crazy story.
Just 5 years before the wall fell.
Yeah, it was exactly. It was while Hungary was still under the communist regime. Mm-hmm. So in any event, she met Dr. Weissman, and Dr. Weissman learned about the fact that she was good at making messenger RNA. And Dr. Carrico was actually working with cardiologists then, and they were hoping that they can use messenger RNA to help develop new types of coronary artery bypass grafts. And that work wasn't successful. But when Dr. Weissman found out about it, she said, okay, well, why don't we make some RNA together and see if we can figure out how that can be useful. So they turned out that together, they came up with the key step that made messenger RNA possible as a therapeutic. And what that was is they changed the base pair of uracil to another form of uracil. And as you can see here, the pseudo-uracil, which is a very minor tweak in the molecule compared with uracil, makes messenger RNA more stable and less likely to cause inflammation. So I like to remind people that messenger RNA, people get worried about putting something that's genetic in their body, but messenger RNA is like the body's email. That's the nuclear material that goes from the nucleus of a cell to the cytoplasm of the cell where proteins are made. And it only survives for a very short period of time. Once that messenger RNA is exposed to the bloodstream, it's broken down very, very quickly. In fact, it's broken down so quickly that they had to change it a little bit to make it a little bit longer lasting. And then the other breakthrough that this pair came up with was the fact that if you put the messenger RNA in these little fat particles, we call them lipid nanoparticles, that you make it stable enough where it can teach, the messenger RNA can teach the muscle cells of your body to make a little bit of the antigen or the protein that you want the immune system to recognize to protect the body. And of course, for COVID-19, that antigen is the spike protein. So with these breakthroughs, Dr. Weissman and Dr. Carrico licensed this concept to BioNTech, which is a German startup company that worked with Pfizer to create the Pfizer vaccine, and with Moderna, which is a company out of Boston that is also involved in the messenger RNA area. In fact, they're so involved in messenger RNA that the RNA is in their name, Moderna. That's why it's called Moderna. So it turns out that this collaboration of somebody who grew up in communist, extremely poor Central Europe, and somebody that is like me, worked out to this would be a tremendous collaboration. And then the other part of the collaboration is Dr. Barney Graham. You can see him on the bottom of the slide. And he was an immunologist that was hired by the US government to set up a vaccine research program. And at that time, the government was actually worried about bioterrorism. And they wanted him to come up with ideas to prevent people from possible bioterrorist attacks. And so he had the insight to know that a coronavirus could be one of those things that could cause a bioterrorist attack. I'm going to just take a quick sip of water here. And he was fortunate to have that insight and to develop a database bank of all the possible configurations of different viruses, including the coronavirus. So when COVID-19 hit, Barney Graham and his team jumped into action. And there's something that's very, very interesting about proteins. So the messenger RNA tells you how to build the protein. But the way the protein folds is an extremely important part of how it works in the body and how it can either have good consequences or bad consequences. So you may have heard of amyloid plaques in Alzheimer's. Well, amyloid plaques is just a protein that folds in a bad way that causes damage. So not only did they need to know what the genetic material is of the spike protein, they needed to know how it folded in order to create a workable vaccine. And Dr. Graham was the one that determined the configuration of the folding so that you can create a vaccine against that particular spike protein. So, interestingly, Dr. Graham he's Caucasian, but he's married to an African-American woman, and he happens to have a team of mostly black scientists. And you can see the team here. So it's interesting. A lot of them actually immigrated from Africa. And so his team was actually responsible for coming up with this configuration for the spike protein, which was essential in terms of creating the vaccine. So you put together the work of Dr. Carrico and Dr. Weissman and Dr. Graham and his team, and you come up with this amazing collaboration, all people from different backgrounds. By the way, Dr. Graham was very different than the others. He grew up on a farm in Kansas, and then eventually got trained in the Midwest and came to the United States. So what's so interesting is that success, in my opinion, is often related to these inputs from people that have different perspectives, this cross-cultural magic. And during the pandemic, there was actually examples of that. You would never know that if you just listened to the politics. But here's a picture of Dr. Graham showing Dr. Fauci and President Trump his finding of the structure of the spike protein. And this literally happened a couple of weeks after discovering the genetics of COVID-19. It's amazing that we went from a virus that was completely unknown to humanity, to knowing its genetic code, to knowing how it's structured, to creating a workable vaccine in 66 days. And usually vaccines took 10 years to develop. So this is truly breakthrough technology. And now we know that these vaccines are incredibly safe and effective. So it's just a remarkable story that I don't think gets told enough. And there's also a local component to this story. So we were fortunate when Pfizer, Moderna, and Novavax and some of the other vaccine manufacturers were looking to do the clinical research for this, they had to reach out to people in the clinical research community. As mentioned, I'm a cardiologist. I have not spent a career in infectious disease. But because I'm an expert in running clinical trials, I was approached by Pfizer and Moderna and Novavax to run the clinical trials here in Jacksonville. And fortunately, we had a great team, and we had a lot of participation amongst patients. And we put in approximately 1,500 patients in COVID-19 vaccine research here in Northeast Florida, which is amazing. And kudos, really, to all the people that make it happen. You know, I'm just a small part of the puzzle, but these are all the people in our organization that were involved in running the vaccine trials here in Jacksonville. So one, we should feel good about it in terms of the fact that, one, we were a big part of this whole international vaccine development here in Northeast Florida, and two, is that we have this tremendous cooperation between people in the community and our research folks in order to make this happen. So I actually want to give you a round of applause for being part of this.
And being here. Thank you.
So, you know, it's a long story, but it really started with an evangelical minister and an African slave. And ironically, those were two groups of people that may have been the most skeptical about vaccines, that actually started the vaccine movement in the US, and that translated through cross-cultural cooperation to the point where we have now, where we have these breakthrough therapies that have literally saved many, many, many lives, and will continue to save lives as we learn how to apply these technologies to other diseases. So I'm gonna hand it back to Vicki to talk about our current vaccine strategies and some of the other diseases we're working on.
So I'm not a cardiologist, but I do deal with vaccines. I was a pediatrician in general practice for about 30 years. I currently work in the emergency room, as well as see some patients in the pediatric practice, and then, of course, do the clinical research. And so I've had lots of exposure to vaccine, worked on vaccine studies back in the late 70s, early 80s. The pneumococcal vaccine that you get, I worked on in the early 80s. And HIB was another big one. So interesting that he points out that not all vaccines are the same. Like he said, that collaboration that resulted in 66 days in a vaccine was this messenger RNA. He likes to call it the email. I like to call it the recipe. Our DNA is our library, and it's in every one of our cells, the entire library of who we are. Each cell only reads some of those books and some of those recipes. And so our muscle cell normally reads the recipe to make a muscle cell. But when you inject a messenger RNA into it, that recipe, that cell says, oh, I'm going to make this instead, and reads the recipe for the spike protein and puts it out there into the bloodstream. The body says, you're not a muscle cell, you're something foreign, and makes some protection and immunity against it. So some of the first vaccines that were around were live attenuated. They were literally taking live pus that had the virus in it and putting it into somebody else. We do it a lot more sophisticatedly now. The measles, mumps, and rubella, and many times the oral polio were actually live viruses, but they were altered so that they couldn't really cause disease, but the body recognized them as foreign. And yes, you do get some of the symptoms even now with the MMR. The children will get a week or up to 14 days later, a rash very similar to measles, sometimes or rubella, which looks very similar to it. I've never seen a response that looks like mumps, but they do get a low-grade fever, et cetera. One of the other ways, are we going to click? Whoops. Is inactivated. It doesn't actually contain a live virus or the bacteria. The polio that we now inject is like that. It's no longer alive that we give it to you orally. It's now injected. It is still the whole virus, though. It's just not alive. And then a subunit where they send the actual protein. So Novavax, which he mentioned we did the studies on, and the Hib and hepatitis B that I mentioned a little while ago, they are actually a protein part of the virus that a body recognizes in the correct configuration, like he said. So figuring out exactly what the recipe was not enough. You also needed to know exactly how to put that recipe together. And anybody who cooks knows sometimes you got to put things in the exact order or it's not going to work out. Same thing with that messenger RNA. Well, this was actually giving that protein, but that little protein is not very stable. So they would hook it together with other things, and that's what we do with the Novavax. And then there is the viral vector, which is similar to what the J&J was. It's similar to not really the inactivated whole virus, but a big part of the virus, hoping to give you an immune response. And those were easiest to make, and J&J was able to quickly make those. The problem is the immunity is not as specific for the spike protein as these messenger RNAs are, nor are they stable, and they're not as effective. And then, of course, the messenger RNA that we have been talking about, that it's a harmless protein to your body. That little spike protein is not going to do anything to your body except tell your body that it doesn't like it. So the next time it sees it, when you inhale that COVID virus from your neighbor, they see it as something foreign, and your body already has an army ready to fight that particular enemy. Sometimes it's going. And so, these viruses are all over. They're ubiquitous, and we actually routinely in the hospital and in the emergency room, as well as during our studies, this is an example of the tests that we test for in our COVID patients. They don't just test for RSV, especially if you come in sick. I mean, not just test for COVID, especially if you come in sick. We want to know what's making you sick. If it's not COVID, what is it? Is it one of these other viruses that can mimic it? And this is a typical nasal swab that we can find this out. And, we've fought these viruses back in the mummy times, and we still continue to, have them, and therefore we need to know how to fight them. And the best way to fight them, of course, is to prevent against them, prevent that war from happening in our body. And the best way to do that is with the vaccines. And, these vaccines change, and so we need to keep on making the new vaccines. So this is actually one that we use in the hospital where I work and is used in the community. We've been testing for coronavirus. As you can see, there's several of them there that we test for. We've been testing for those for 15 years.
Yeah, it's amazing. We can learn about these things literally within a few minutes. Mm-hmm. We never had that capability before.
Right. And so, I've told a lot of people RSV is actually one of the studies you'll see in a moment that we are working on an RSV vaccine. And many of us have had friends that, have an illness and they're in the hospital for something completely unrelated, maybe a cancer, maybe they were in the hospital from a trauma or surgery, and they get pneumonia and they die from this pneumonia. Well, a lot of times we're now learning, because of these nasal swabs, that it's RSV that causes a simple runny nose in most people. We know it causes horrible disease in little babies, and so we've routinely checked little babies. But now, because this is part of our routine COVID testing in the ER and in the hospital, lo and behold, we're finding these viruses causing similar diseases and destruction that COVID can cause, the most important of which is RSV. And like I said, we are doing a trial with RSV vaccine in 60 and over, because that's the age group that suffers with it, as well as little babies, hoping, of course, from my standpoint as a pediatrician, we can get it to our babies next. And so, like I said, we are doing plenty of the studies. We've already done all of the COVID, Moderna, we've done the Pfizer and the Novavax. We're actually branching out to a Sanofi pediatric COVID vaccine, a little different methodology, not messenger RNA, but this messenger RNA booster right now we have with an Omicron variant. And for 12 and up, we have chikungunya vaccine, which is...
Yeah, just the Omicron variant vaccines are not commercially available. You can only get them through research.
Right, through research, yeah. The chikungunya vaccine, which is a disease that is quite debilitating, was primarily in Africa and South America. And Miami, Miami is just being wrought with it right now. My son was a student, medical field student down in Miami, and saw a child who had chikungunya. One of our lectures here, we had a participant that had chikungunya walking with a cane all contorted up, because unlike polio, this one contorts your body up. and is now becoming a huge issue in America. So we do have a vaccine trial going on with that one. Then, like I talked about, the messenger RNA for adults at this time, we'd like to get it to our children. And we're working also on an RSV flu vaccine combination because you do have to get the flu every year. We suspect with COVID and RSV, you're going to have to get that regularly as well. But the two of them go together nicely, and we have a study for that. And like I said, the pediatric COVID vaccine, that's a protein one, bound, like I said, the protein bound in the old-fashioned Hib vaccines, et cetera, not a messenger RNA. There's been some people who really, in the media, of course, that don't understand that messenger RNA has nothing to do with your DNA, but this is just another way, if that is something that concerns you, that you can rest assured that that vaccine's a little bit different. And so with that said, the only way we can do all of this research is with volunteers. And obviously all of you, most of you had received emails, so you already are aware of us. So we need you to tell 2 friends that you're great contributors to getting this done, whether it's the 6 volunteers in this prison who got a great opportunity
get
to
out of
get
jail
off.
free card.
Yeah, except for, 3 of them ended up back, one of which, the reason I said talk about his wig, was one of them had stolen a wig, and that's why he was there. His sentence was deportation, and he returned, and that's why he was sentenced to death. And then he went out and sold a wig again. Anyway.
Well, I didn't make the wig comment in the interest of time because
Okay.
I realized running behind, but if somebody asks a question about it, I will talk about it.
Anyway, so we can educate you on your health, nurture you with personalized care. We don't take the place of your primary care doctor, but it is another set of physicians caring for you. You are compensated for these studies and getting cutting-edge treatments. Like he said, the Omicron is not available in the community. It's only available in a research capacity. Many of the medications now are only available in a research capacity, but you get those. And you offer something to your whole community and society by being one of those that ends up in one of the New England Journal reports of being one of those pieces of data. And then it lets you know even more about your health and take better control. Company's named ENCORE. There you go, ENCORE Docs. That's the phone number, and we have lots of information on the tables.